The Importance of Natural Products in Biochemistry and Medicine by Mira,
Michelle,
and Melissa.
Part 1. Historical Importance of Natural Products. Historically, natural products have been used since ancient times and in folklore for the treatment of many diseases and illnesses. The earliest records of natural products were depicted on clay tablets in cuneiform from Mesopotamia, which documented oils and myrrh, which are still used today to treat coughs, colds, and inflammation. The Ebis Papyrus is an Egyptian pharmaceutical record which documents over 700 plant-based drugs ranging from gargles, pills, infusions, to ointments. The Greek physician Dioscorides recorded the collection, storage, and the uses of medicinal herbs, while the Greek philosopher and natural scientist Theophrastus dealt with medicinal herbs. It was the Arabs who were the first to privately own pharmacies, with Avicenna, a Persian pharmacist, physician, philosopher, and poet, contributing much to the sciences of pharmacy and medicine. Needless to say, natural products have had a powerful influence on the medical and scientific world. Some of the most important modern drugs are natural products. Probably the most famous and well-known example to date would be the synthesis of the anti-inflammatory agent acetylsalicylic acid, derived from the natural product salicin, isolated from the bark of the willow tree. Investigation of opium poppy resulted in the isolation of several alkaloids, including morphine, a commercially important drug. The anti-malarial drug quinine, approved by the United States FDA in 2004, isolated from the bark of the Cinchona succirubra, had been used for centuries for the treatment of malaria, fever, indigestion, mouth and throat diseases, and cancer.
Part 2. Applications of natural products in cancer research. Human cancer is associated with alterations in a number of oncogenes and tumor suppressor genes that occur at various stages, from carcinogenesis to tumor growth, progression, and metastasis. Oncogenes normally control the process that lead a cell to become cancerous, like gain-of-function mutations, amplification, and overexpression. In contrast, tumor suppressor genes are good genes that can slow cell division and growth. So the critical balance between oncogenes and tumor suppressor genes controls the initiation and progression of cancer. Molecules in natural products have shown the ability to regulate oncogenes in the same way that tumor suppressor genes do. The p53-MDM2 pathway is a significant pathway in cancer formation. MDM2 is the oncogene that causes cells to become cancerous, and the p53 gene is the tumor suppressor gene that targets it. p53 forms a negative feedback loop with MDM2, which directly interacts with p53 and inhibits its function and expression. Many natural products target the p53-MDM2 pathway, including chalcones, genistein, curcumin, sesquiterpenoids, and ginsenosides. More plants that can target this pathway are actively being discovered, and they're all derived from plants.
Part 3. Current research. Recent work in cancer research has led to the discovery of a family of drugs called camptothecins. Wall and Wani first isolated 20S camptothecin in 1966 from the bark of Camptotheca acuminata, but observed that this compound had poor solubility and stability. It was discovered just a year shy before they discovered another anti-cancer drug, paclitaxel, which soon became a broad-spectrum chemotherapeutic. However, interest in camptothecin picked up in the late 80s when a molecular target for it was found, topoisomerases. Topoisomerases are a class of enzymes that facilitate DNA unwinding during transcription and replication by relaxing DNA supercoils. Mechanistically, these drugs inhibit DNA topoisomerases from repairing their induced breaks in the DNA and ultimately convert these essential enzymes into DNA-damaging agents. The presence of these inhibitors can prevent rapid cell proliferation, thus being ideal drugs to target cancer cells or bacteria. With optimism to inhibit topoisomerase activity in cancerous cells, various analogs were synthesized in an attempt to overcome the solubility and stability issues. Currently, 2 analogs are used in practice for cancer chemotherapy: irinotecan, used for metastatic colorectal cancer, and topotecan, which has been approved for ovarian, cervical cancer, and small cell lung cancer. Drug development of these inhibitors has posed many difficulties in initial identification, design, and synthesis of novel topoisomerase targeting molecules due to their selectivity problems and continues in modern research. However, this should not deter us away from the quest to discover the number of natural products that can potentially inhibit topoisomerases, as well as other molecular targets that can prevent other cancer pathways. The vast majority of natural products have not yet been found, and an abundance of novel and exciting chemistry and biology awaits discovery. Thousands of natural products have been synthesized, which once looked like an impossible task only until recently.